for the DNA fingerprinting, for example, you would target the area in our genome that is different, that has more variation. You don't want to target like the hemoglobin gene where it's all exactly the same for everybody. We're not going to be able to tell the DNA apart from each other. And then if you're trying to target a gene, like say the PGLO gene for transformation, once again, you're just aiming for a certain spot. And that's what you copy over and over and over again. And actually, when you do that, you put all the DNA in the tube, but you only target that spot. So that's what gets copied and copied and copied. So it's also a way to isolate that, because the rest is not copied. Yeah, it's in there, but you have made sometimes billions, but you've made millions for sure of the copies of your gene, and there's only, what you started with in the test tube. It was never copied. So then it's easy to pull out the stuff that there's a lot of. Okay, so how we cut DNA is restriction enzymes. Now, do you remember a little bit about restriction enzymes from doing the virtual lab? Did we talk about them much, what they do? A little bit? Okay, so bacteria make these enzymes that cut DNA. And they are called restriction enzymes because they're restricted to cutting only at a certain spot. That's the code, the sequence of A, T, C's, G's that they cut at. And bacteria have these restriction enzymes because they are constantly being bombarded with foreign DNA. So if they cut up the DNA of a virus or another bacteria, then they're not wasting time making the enzymes or the parts for those invaders. Okay, so then when we do DNA fingerprinting, we use a couple different restriction enzymes. Victoria, can you shut that and listen to me pretty please? So we put a few different enzymes in the tube so that then your sequence is cut up in different ways, so then you're going to get more pieces. So for example, enzyme number 1 on the sample of DNA, it had 4 sites with the exact same code, so it cut it 4 times, making how many fragments? Since it's a line, when it cuts something 4 times, how many fragments does it make? 5. Now with a different enzyme, it only found its specific code, notice, on the exact same sequence of DNA, they color-coded it to show you that. Its restriction site only happened 3 places. So now we have all these different fragment sizes in the test tube, and they're going to spread out right by size on the gel. But that's for DNA fingerprinting, but again, if we're trying to cut out a certain gene, then we have to look for as an enzyme that's going to cut right in front of our gene. And sometimes we can find them, sometimes we can't. Okay, so here's the PCR machine. So this is what makes the copies. It's called polymerase chain reaction because it's a chain of reactions. It repeats and repeats and repeats like links in a chain. So you guys know a little, but you know a lot, hopefully, about DNA replication, right? The double strand has to unzip. Those hydrogen bonds have to break. Once they break, the DNA polymerase attaches. Remember when you guys did the pop bead lab? And you made the other side. Well, scientists realized if you just heat DNA up, because the hydrogen bonds are weak, it splits apart. And it doesn't want to be split apart. So then you throw in there the enzyme, the DNA polymerase, into the tube. You throw in A's, T's, C's, and G's, your bases. You start letting it cool down, and it automatically starts to bond to those pieces because it wants to be a double strand. The A wants to bond to a T, okay, and a C wants to bond to a G. We put a little primer in there that is going to target it. So we know the sequence right in front of our gene, so we build a little piece of DNA, and it is attracted to there, and then that starts the sequence, starts the replication right behind it. And so then we keep making these copies, and then here's one cycle, like a chain of reactions. So we've doubled it, another cycle, we've doubled it again, and like one of the PCRs I used to have to do for the pseudoscorpions took 33 cycles. Okay, so you're going to get just tons and tons and tons of copies of the area I was targeting. The other thing PCR is useful for, instead of just making these areas that we want to, is if you don't have much of a DNA sample, you can get the PCR to copy your sample that way also. So that way this lab can have some and that lab, and you guys know how criminal cases go, right? Defense needs samples. The bad guys, oh no, that's defense. What's the guys that are the prosecutors? They need samples, right? The police department, you know, they have samples. So this stops people from fighting over the samples too. We can make copies. Okay, this one is really a review, you guys. But if you notice, this chapter is more using our information in a practical sense. So mutations, you guys should know what they are. Changes in the DNA sequence. They're caused from certain chemicals and radiation. What are those things called that cause mutations? Mutagens, good. And if the mutation causes cancer, what do we call it? A carcinogen. So, right, these things are actually changing the DNA. Most mutations do happen when the DNA is copied. And then there's those different types. Again, you should have seen these words. Point mutations like sickle cell anemia. And this is bringing, this information should be coming back to when, if you did a gene mutation for your little mini research paper. This is what we're talking about. So one base has been messed up, has changed. It should have been a T, now it's a or whatever it is, okay? Now, missense, you guys remember, are types of point mutations, and they actually change the protein. That's what happens in sickle cell anemia. Then also nonsense, where now the code was changed, so the messenger RNA made a stop codon. Remember when we sat over here and we played a little bingo game? So if all of a sudden a codon, because of a mutation, said to stop building the protein, now we will have a really big problem, right? Because we just severed the protein. It's not going to be functional. So that's even worse. They call that nonsense. Okay? Then frame shift mutations we've also talked about. That is where you had an insertion or a deletion, and it shifted everything over. I used the analogy of your guys' scantrons or bubble maps, if you got off one. Right, so check this out. The red cat ate the fat rat. So we're trying to show you an example here that you're really familiar with words and how they work. And so we use three-letter words to compare to the codons, right, for messenger RNA. So if it's a point mutation, if it's missense, we had a substitution, and it changed one word. The red hat ate the fat rat. And then you have the effect on the DNA showing one change, and the effect on which amino acid was put into place. Then the nonsense again, we got rid of a word. The red cat ate the fat, and we don't get to know the rest of the story. Okay, now here's frame shift, you guys. Addition. So here is the web. A B was slammed in here, changing the words all the way down so they don't make sense at all. So that's an insertion or an addition where the DNA polymerase just put an extra one in there. It doesn't have a place to bond, but it just shifted all the way over when the messenger RNA was being read. Deletion, so now we lost the D, so the C shifts over. The rec, ada, teat, hef, doesn't make any sense. Just like the protein's not going to work. Okay, translocation. So this is one of the newer words that you might not have noticed. So back in meiosis, you have the matching chromosomes finding each other. That was called synapse. Do you guys remember when we did the modeling? And I had you guys do what? When you found your homologs, what happened between the homologs? They traded pieces to mix themselves up. Well, when you guys were doing the modeling, was there sometimes other chromosomes kind of in your way and kind of too close to you? So sometimes the different chromosomes could trade information with each other, but that's not good. Okay, because now on one of them, you have information that's not supposed to be there, and same thing on the other one. And that's called translocation. So now genes have translocated to a different chromosome that they're not supposed to be on. And they're not going to work as well. Okay, 9 and 22, we found examples where this happens sometimes. And we name the chromosome that's a combination, the little one at least, that causes more problems, the Philadelphia chromosome. So that's translocation. Is that a gene mutation? Or chromosomal mutation. So this is how, remember, if you did a chromosomal disorder, they said that your disorder might have been caused from an extra chromosome or a missing chromosome or an altered chromosome, right? Or a chromosome was messed up, however, whatever words they used. That happens to crossing over. Okay, so then how can we fix our genes, basically, is gene therapy. And gene therapy, fixes the code so that the correct proteins are being made. Instead of us just treating the symptoms, right? We're changing the DNA or we're adding genes. For the Power of Genes video, do you guys remember that the guy that had the angina, he had the major heart disease, he needed more vessels in his heart to grow? Do you guys remember that one? Did it kind of work? Yeah, so they inserted, remember they had like a little syringe in his heart, and you saw the doctor, it wasn't that big. The doctor pushed it in through a tube. Do you guys remember that? So what he pushed in there was plasmids, or I'd have to look it up. It could have been viruses. Viruses that we have changed so they're just carrying DNA and inserting the DNA into his heart cells. I think normally with eukaryotic cells we use viruses to get the DNA inside the cells. With bacteria we use the plasmids more regularly. I'd have to research which one because they didn't tell us, I don't think, did they? Okay, but anyway, the idea is that this is gene therapy. At the bottom one here, this is treats somatic cells. So they put genes in the cells, and those genes were being expressed, and the correct proteins were being made that helped rebuild vessels, and it worked. But pretty soon, I think those foreign genes kind of get cleared out. So he maybe would have to do it a few times. But who cares? If it worked for a while and he got new healthy blood vessels, a few of them, then he was kind of cured, right? Okay, now the other type is more like Gattaca. Now we're going to change the genes of like a zygote. Okay, we're going to clone things. That would be a type of gene therapy where we're looking at embryos, but we're talking like when they're just a blastula, just a ball of little cells. That's all they are. There's no differentiation at all. And if we were to give these cells a new gene to express, it hopefully would get incorporated into the chromosome, and then they would have the gene. Okay? A lot of that is still a little bit of science fiction right now, but it's getting much closer. And I think in London, or England, London's the capital, they approved doing something like this with one particular disease. What is happening is not gene therapy, is more of an observational thing in many countries where, say, a person knows, two parents know that they have 50% chance, or maybe it's even higher than that, of passing on a certain trait to their offspring. They can do in vitro, right, where they have the little blastulas in the test tubes or in the petri dishes, and they can look at them. And they could pluck one cell away from it, extract the DNA, and see if the DNA has the particular bad gene. Or, hey, did this little blastula right here have the healthy gene? Okay, let's only implant these blastulas. They only have the healthy gene. We haven't engineered them. We just spied on them when we normally couldn't see what's going on. And then we did in vitro. Some people still might be uncomfortable with this much interaction and choosing, okay? But that's what we can do right now. That's a little different than manipulation, though. Okay, almost done here, you guys. So this is what I talked about. There's also a way that we can put DNA in these little lipid vesicles, like little cells, and get another cell to incorporate that. There is a lot of success going on with cystic fibrosis. Okay, do you remember that? That's when a Nurse Story talked about it with us, right? Where they have to pound their chest because they make way too much. I want to say phlegm, but that's not necessarily the right word. Mucus. Thank you. They're getting very successful doing some gene therapy for those individuals. Okay, then it gets to the ethics thing. If you guys think we should be doing this or not. But one thing is, it does also give us information to make choices. Okay, see if you guys can do that.